video is about enzymes. Enzymes are special proteins whose job it is to put molecules together. Maybe they break molecules apart, and sometimes they catalyze reactions, which just means they make them go faster. Enzymes, these special proteins, it's all about their structure. Their structure, their shape, is everything because it's what's going to determine what the enzyme can do. So here is my enzyme here, and so its structure is like a Pac-Man, and it is perfectly shaped to break down this triangular substrate. A substrate is just anything that an enzyme is acting on. And there's a very special part of this enzyme called the active site, which is the part of the protein that's actually going to be doing the breaking down or putting together. So when an enzyme and a substrate join together at the active site, they form an enzyme-substrate complex. So when the substrate is in the active site of the enzyme, it puts strain on the substrate, and then the substrate breaks apart. So then you're left still with the enzyme. The enzyme doesn't get used up. So you've got the enzyme and finally the products that are a result of breaking down the substrate. What determines enzyme's special structure is the interactions between the amino acid side chains that make up that protein. So as you recall, the proteins are made up of a string of amino acids that are connected by peptide bonds, and each of those amino acids has its own side chain that has a special property. So this is my example of a tripeptide here, and these different amino acids, side chains I have in different colors because we're going to imagine they have different properties. So some side chains are hydrophobic, meaning they don't like to be around water. Some, conversely, are hydrophilic, meaning they like to be around water. Some are negatively charged, and some are positively charged. So, negative and positively charged amino acids, they like to be near each other in the folding of a protein. And some also contain sulfur, and there are special cases, but it's good to know just that sulfurs like each other, and they will pair up together when a protein is folding. So here in my example, we can see that before I had this linear polypeptide, but because this one was positively charged and this one was negatively charged, they came together, so you can actually see this kind of bending around, and our positively and negatively charged ones are now close together. And since the middle one was hydrophilic, meaning it likes water, we're assuming that this is like a water environment, for example, the cytoplasm of a cell. That side chain is going to move towards the outside in order to be close to water. If it were hydrophobic, it would have wanted to stay inside and away from water. Enzyme structure are also particularly built depending on their environment. So they're suited to very specific temperature and pH conditions. So enzymes can work very poorly, or they can work very well. Right? Enzymes that work very well are making a whole lot of products. Ones that have low enzyme activity are not making very many products. And let's look at why. So if we're looking at temperature, enzymes will, at low temperatures, typically have low enzyme activity. Okay? But as that temperature increases, right, the energy of the molecules increases, and the substrates and the enzymes start running into each other more, and so more and more products can be made because the molecules are moving faster. And they will, move faster and be more effective until it gets to a certain optimum temperature. For example, in your body temperature is 98.6 degrees, so it's probably reasonable to think that most of the enzymes in your body have an optimum temperature very close to that. But if the temperature starts to get too high, then those molecules that were like moving really nice start going everywhere, and it actually breaks some of the bonds that have been holding that enzyme together in its special structure. Same with pH. There's an optimum pH, or this certain like amount of acidity or basicity that an enzyme is in that makes it work the best. So if an enzyme is in a too acidic environment, if it's too acidic, then the negatively charged amino acids are going to be attracted to the hydrogen ions on the outside of it. And so instead of being folded in, to make this special structure, some will want to leave and go over here or over here where all of the oppositely charged ions are. So if it's too acidic, negatively charged amino acids will be attracted to the H+ ions. If it's in a too basic environment, then the positively charged amino acids are going to be attracted to the hydroxide ions. So that's going to cause a difference in folding, which of course then, if your structure is mutated, then your function is not going to be able to happen. Here's my little diagram. And the super sassy science name for the shape changing so much that it can't do its job anymore is called denaturing. So if the environment is too acidic, too basic, or too hot, the enzyme will actually denature. Bonds will break, and the enzyme will come apart. Now, if it's too cold, the enzyme's not going to break, it's just going to be moving so slowly that not a whole lot of interaction between enzymes and substrates are going to happen. So if your protein denatures, so let's imagine that my enzyme still looks like a Pac-Man here, but then the temperature increases, right, and it starts moving too fast, and then its shape has changed so much that the active site, it's all wiggly, and it can no longer accept the substrate, which is that green triangle if you remember from before. And therefore, since our enzyme can't act on our substrate, we're not going to get any of the products that we were seeking.